Today, I'd like to talk about the easy-to-understanding genetic testing takes the first step towards diagnosis. So, our body is made by cells, and each cell containing the nucleus. In the nucleus, we have the DNA. And protein are a component of our body are synthesized based on the DNA information. And genetic information in DNA is used to synthesize protein in two steps. First, DNA is transcribed into the messenger RNA like this, which carries the genetic information out of the nucleus. And then the ribosome reads the messenger RNA sequence and translates it into the protein, a functional and structural component of our body. So why genetic hearing loss occurs? Proteins are synthesized based on the DNA information, as I mentioned in the previous slide. DNA without a pathogenic variant, functional protein, normal function protein is produced, and thus we can hear normally. However, in situation with a pathogenic variant like this, in the inner ear component gene, the messenger RNA produced from this gene with pathogenic variant, also containing the pathogenic variant even in the messenger RNA. And based on this messenger RNA, the protein loses the function due to the missense or three-dimensional change, or no protein are produced at all. Thus, we call the hearing, patient causes hearing impairment. So, how many genes are involved in the hearing loss? This graph shows the number of causative genes and identified years. In 1995, first gene, PAU3F4, is identified. And now we identified over 150 genes have been reported as causative for non-syndromic hearing loss. So, in genetic testing for hearing loss patient, we should analyze over 150 genes. Thus, we need the efficient genetic analysis methods. So, genetic testing methods for hearing loss is also developed. Till the 2008, Sanger sequencing is mainly used. This analyzing the genes one by one manner. So it takes so many times and so many costs. So we changed to the embed assay, analyzing the commonly observed pathogenic variants simultaneously. And now we use the next generation sequencer from the 2015. It is possible to analyze all deafness causative genes comprehensively. Okay. So what is the next generation sequencer? This graph shows the sequencing machine launched year and the number of the bases we can read within 1 day. Okay, these three is a conventional Sanger sequencer, and this is a next generation sequencer. There are several companies selling several machines of next generation sequencer. And as you can see, the total amount of produced or read, possible to read DNA sequencer is 10-fold every 2 years, increasing. Next generation sequencer is a machine that determines a huge number of DNA sequencing by performing sequencing reaction in massively parallel manner. For example, 80 million or 10,000 million parallel sequencing reaction are performed at the same time. So we can get huge number of the data. So NGS enables us to comprehensive genetic testing for all known deafness genes. This is a simple comparison to the Sanger sequencer and the next generation sequencer. Sanger sequencer is used in the Human Genome Project, And to determine one person genome sequence, it requires 13 years, and it requires $30,000 million US per person. And now we can access the next generation sequencer. It is possible to determine 160-person genome at the same time within 10 days. And the cost is only $800 US per person. So next generation sequencing is very, very powerful tool for analyzing the genetic analysis of hearing loss. So there are three main strategies to determine the genetic cause of hearing loss. First one is a target gene sequencing of all known deafness genes. As I mentioned, up to now, we identified almost 150 genes for the genetic cause of deafness. So we sequence all deafness genes, only the all deafness genes. This is the first strategy. Second strategy is the whole exome sequencing, analyzing whole genes, about 23,000 genes at the same time. And last one is the whole genome sequencing, analyzing all DNA sequencing of the patient. And now, target sequencing, only analyzing the deafness gene, is a useful and cost-effective analysis method for the deafness. And very important thing of the next generation sequencing analysis, we identified so many variants, but all variants are not the genetic cause of hearing loss. In human DNA, only the 3% of region is protein coding genes. And every people have different DNA sequence, about 1 out of 1,000 bases. Variants in non-coding region, this A and B are gene, and this white region is a non-coding region without gene, is usually non-pathogenic. And even within the gene A, variant genes that retain the protein function is not affect the hearing function, so they are normal hearing. However, in the variant in gene A deteriorates the protein function, finally causes hearing loss. So it is important to figure out which one is the genetic cause of the identified variant. When we analyzed 158 deafness genes for one hearing loss patient, we identified about 900 variants per person. However, a genetic cause of each patient is 1 or 2 variants. So the restrict or filtering procedure is very, very important to get the final conclusion. First, we select the variant estimated to have no effect on protein function are removed. And then we remove the variant commonly observed in normal hearing control. That is without any pathogenic effects. And to reach a final conclusion, we also use the family segregation analysis, family history of affected patient, and phenotype information. Type of hearing loss or severity hearing loss information is very, very important to reach a final conclusion. And we also use a large-scale database. We've already analyzing the 20,000 patients. So we identified about 1,000 variants from one patient, but responsible variant for hearing loss is 1 or 2. So appropriate filtering and pathogenicity interpretation is a key issue for accurate diagnosis. Thank you very much.
Thank you.